In this video, we're going to talk about protein synthesis, and specifically, we're going to focus on the differences between DNA and RNA and talk a little bit more about transcription and translation. All right, so the first thing that we need to understand is the difference between DNA and RNA. DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. And here's a diagram showing the difference. So the first difference is that DNA is a double-stranded or double helix structure, and RNA is single-stranded. The second big difference is that RNA is going to contain a sugar known as ribose. In DNA, the sugar is deoxyribose. And a third major difference between the two is that RNA has a uracil instead of thymine. So that means that the bonding rules have changed. A goes to U when we're making RNA, and G goes to C. So there's no thymine in RNA. There are three types of RNA that we need to be familiar with. The first is what's known as messenger RNA, and this is the RNA molecule that's responsible for carrying the message from the nucleus, or from the DNA, to the ribosome. We have ribosomal RNA, which is the RNA that makes up the ribosome, which remembers the organelle responsible for protein synthesis. And then we have this transfer RNA, which is responsible for carrying amino acids, the monomers of proteins, to the ribosome to aid in protein synthesis. This whole concept leads us to something called the central dogma of molecular biology. And this central dogma, or this main idea, is this kind of flowchart here going from DNA to RNA, and the RNA coding for a specific protein. The proteins are responsible for all of your traits and everything that occurs in your body. And going from this process, we have transcription and translation, which is what we're going to focus on in this video. So let's talk about transcription. This is the process of going from DNA to a messenger RNA. And this is going to kind of occur in three main steps. The first step is that the DNA molecule is going to unzip or unwind, very similar to what we talked about with DNA replication. RNA polymerase is going to come in, and it's going to attach to a promoter region, also known as the Tata box. It's called the Tata box because it's a series of thymines and adenines that don't code for anything. And so the RNA polymerase knows to bind there in that region. And it's going to add RNA nucleotides. So it's going to add in A, T, G, or A, U's, G's, and C's. Remember, there's no T's. And just like DNA polymerase, it's always going to add in a 5' to 3' direction, meaning it's always going to be attaching to that free hydroxide group. At the end of transcription, you have what's called a pre-mRNA transcript, which has to be processed. So what does it mean to process something? Well, it means to make edits. And so there are two main parts of that mRNA transcript. You have exons and introns. Exons are the protein coding regions, meaning they are the important regions that code for something. They are going to exit the nucleus to go to the ribosome. Introns do not code for anything, so they're non-coding or non-protein coding regions. So they're going to stay inside the nucleus because we don't want to do anything with them. These introns are going to be removed by an enzyme known as ribozyme, and after those introns are removed, we're going to add a GTP cap to the 5' end. GTP stands for guanosine triphosphate. It has a very similar function to ATP and structure. And then we're also going to add a poly A tail, or in other words, we're going to add several adenines bonded together to the 3' end. This is just a diagram showing you that. So here we have our DNA strand. mRNA is made from that through transcription, which contains introns, or those non-coding regions. We're going to splice those out by ribozyme, and then we're going to add in a cap to the 5' and a bunch of A's to the 3'. So after our processing has occurred, we have to go into translation. And translation is the process of taking that mRNA and turning it into a protein. And this is going to occur in three main steps. You have initiation, elongation, and then termination. So we have three diagrams here we're going to walk through. This first diagram is talking about initiation. So what happens is that mRNA that has been processed leaves the nucleus and goes out into the cytoplasm. You have a small ribosomal subunit. So it's the small part of a ribosome that attaches to that mRNA strand. And we also have a large subunit that comes and covers that strand as well. So we've now assembled a ribosome. In addition, you have this tRNA molecule that comes in. Remember, tRNA carries amino acids. It's going to carry the start amino acid, methionine. It's going to plug in there to the start codon. A codon is a series of 3 nitrogen bases, and we now are ready for protein synthesis to occur. So now we have elongation. Well, what happens is those tRNA molecules, they are composed of an amino acid, the RNA part, and then at the very end they have what's called an anticodon, or the 3 letters that match up to the codons on the mRNA strand. Each tRNA molecule is going to come into that ribosome and attach, hooking together those amino acids. And so it's going to look very similar to an assembly line. So we start off with methionine. The next tRNA molecule will carry the next amino acid, which could be something like lysine or glycine or cytosine. And that's going to continue as we grow a polypeptide chain, which is a series of amino acids. This is going to occur until we reach a stop codon. Remember again, a codon is a series of 3 nitrogen bases on mRNA. So then we have termination. This is when that stop codon is reached. And there are 3 stop codons, UAG, UAA, and UGA. When that happens, a release factor is going to come into the ribosome, telling the ribosome that it's done working with the mRNA strand. It's going to release the polypeptide chain, or that chain of amino acids. The ribosome pulls apart, and that sequence of amino acids will then go through the Golgi apparatus to finish being folded and sent out to do whatever it needs to do. So translation is a pretty complicated process, but remember, it's 3 main parts. Some of the terms you need to remember: a codon, which is 3 letters on an mRNA strand, and an anticodon, which are the 3 letters at the end of a tRNA, which kind of help the tRNA match up on the mRNA strand. I will post an animation on Moodle. Please go and watch that animation, and I will answer any questions you have tomorrow in class.